In a powerful show of support for nature, nearly every government in the world united behind the CBD's sweeping biodiversity plan for life on Earth. They committed to halting the extinction of threatened animal, plant, and fungi species. And in an historic first, they also committed to helping those species recover, so that the life we share our planet with can not only survive, but thrive. Making these goals a reality will take all of us working together. Reverse the Red is supporting governments, conservation experts, and communities around the world as they join forces to activate tried and tested tools and tie global efforts to local expertise. All in a coordinated effort to assess, plan, and act for wildlife. Together, we can save species from extinction. Together, we can secure our planet's future. Together, we can reverse the red.
Good morning, good afternoon, good evening, good night, wherever you may find yourself in the world. We are very thankful that you have joined us for this incredible webinar. This is part of our series within Reverse the Red on our road to the IUCN World Conservation Congress. So we're particularly excited for this conversation on conservation genetic diversity. There's an amazing group of people that are going to share brilliant thoughts, all sorts of fantastic ideas in kind of strategies on how we think about this and its role within the greater sphere of conservation. So for those of you who may not be as familiar with Reverse the Red, we are a coalition committed to action for threatened species. So our aim is to see that we halt extinctions, reverse declines, and ultimately recover threatened population species. And our coalition is made up of a wide variety of different types of organizations and people. So we have governments, which some of you'll hear from tonight. We have NGOs, we have specialist groups, we have zoos, botanic gardens, aquariums, natural history museums, and more. The commonality is we're all people who are committing actions with the intent of helping species recover. So before turning it over to this incredible discussion, just a few things if you're thinking about getting involved, or a few things to know about. One is the Reverse the Red Species Pledge, which is an international initiative to understand who is working on what actions for specific species. And what that does is help us to coordinate and learn from one another, as well as helping countries and supporting them as they work to deliver on things like Target 4 and Goal A of the Global Biodiversity Framework. And the other thing we have coming up at the IUCN World Conservation Congress is the Reverse the Red Species Pavilion. We've got an incredible set of discussions and cross-cutting discussions that we'll have there. So if you're planning on being at the Congress, please plan to stop by, join some of the sessions, and come and find us there. But without further ado, I would love to turn it over and we'll dig into this fantastic conversation. So Guillaume, it's all yours.
Thank you, Michael. Hello, everyone. Thank you for those nice words and this opportunity. Let me share my screen. Here we go. Yeah, so. Et voilà. Yes, my name is Guillaume Gigot. I work for the French Biodiversity Agency and also the Natural History Museum in Paris. And today we're going to talk about conserving genetic diversity using population and phylogenetic indicators. scaling up resilient species conservation nationally. And actually, when it comes to conservation and public policy, we constantly need charts, indicators, reports, lists. We love lists, species, habitats. They're all interesting guidance tool for decision making. So today, we've built this session to explain how to implement and contribute implementing the coming Montreal Global Biodiversity Framework and contribute to conservation with a pair of those indicators. You will see that the upcoming presentations aim to explain concepts and highlight national case studies for decision-making approach based on genetic diversity indicators, that at different levels. Genetic diversity provides foundations for adaptation and resilience for species, populations, and ecosystems functionality in a rapid changing world. So in the goal A of the Global Biodiversity Framework, you find the Red List Index based on the well-known Red List of the IUCN. And Red List indicators assesses species extinction risk. We encourage, of course, all of us to every country to establish national red lists and to contribute in some way to global assessments too. Here, we will go beyond the red list, also in the line with the goal A of the GBF, and we look at ways of building strategies to conserve as much representative diversity as possible at global and national scales. You will hear about general concepts and national applications for conserving species and their inherent genetic diversity from populations to entire branches of the tree of life. And from where I stand in a public agency and near policymakers, genetic diversity seems to often neglected element. Usually we hear we don't know where to start, how to deal with it, but it is a vital element for long-term conservation actions. So here is the sequence of the different speakers and topics we're going to go through. Thanks to all the speakers who accepted this invitation. And we'll talk about those indicators first at the population level and then at the level of the tree of life. Just a few words. Any 500 indicator measures the proportion of populations with enough genetic diversity and adaptive potential, while EDGE index monitors the conservation status of the most evolutionary distinct and globally endangered species. So I will leave the ground to my colleagues and friends to talk about that. And if you want, Alicia, you can start. You are working at the Royal Botanic Gardens Kew, and you'll start with any 500 indicator, please.
Thank you. Oh, you have
yeah,
to
let
stop.
me, yeah.
Okay.
Here you go.
Okay. Can you confirm if you can see my screen?
Perfect.
Excellent. So welcome. Hello, everyone. I am Alicia Mastreta Llanes. I work at Kew. Today, I will be speaking about the genetic diversity indicators that we have been pushing with a team of people across the world. I also will be sharing some of the results from Jessica da Silva from Zambia, who originally was requested to give this seminar, but she was not able to join. So, question for her part, I will do my best to answer. I would like to start by talking about what is genetic diversity. The first thing is that genetic diversity is part of the beauty of nature. A panther and a jaguar are individuals of the same species, just with different genetic variants related to coat color. Genetic diversity is also to have options because, as you can see in this example of a kelp forest, populations that have more genetic diversity were better able to survive after a heat wave than populations that have less genetic diversity. But what is genetic diversity? Genetic diversity are all of the inheritable variants on the genome of individuals that make individuals of the same species different, but also that make populations of the same species differentiated, maybe because some are part of more colder or warmer parts of the distribution of the species. And then all of the species that form all of the ecosystems have genetic diversity within them. So it's a lot of diversity. Genetic diversity is now part of a global biodiversity framework. As Jerome said, it's part of Goal A and Target 4. I want to highlight that this includes maintaining the adaptive potential of all species, both wild and domesticated. And that, as it is mentioned in Target 4, that this involves conserving genetic diversity both within and between populations. It also means that we need to monitor genetic diversity. To monitor is to measure something, and when you think about genetic diversity, you are probably thinking DNA sequencing, and that can become very expensive very quickly, especially for megadiverse countries that don't have a lot of infrastructure for research. But maybe there's other ways to monitor genetic diversity. And thinking on that, acknowledging how hard it can be to work with genetic diversity, we came up with 2 genetic diversity indicators that focus on processes that affect genetic diversity rather than only relying on DNA data. For that, we ask at the population level the following 2 key questions. The first question is the population still there? Because whenever a population gets extinct, we lose whatever genetic diversity it had, and we don't need genetic data to know that. The second question is if the remaining populations are large enough to maintain genetic diversity, because when populations became too small, they start to lose genetic diversity by something called genetic drift, which is a way of bad luck used by the random processes. We then translate these questions into indicators. The one of the populations is the proportion of populations maintained within a species. This indicator was adopted as a complementary indicator. And as you can see, it's a proportion, therefore it ranges from 0 to 1, with 1 being 100% of populations still remaining. Then for the indicator measuring diversity within populations, we developed the indicator, which was adopted as headline indicator of the proportion of populations within a species that have an effective population size of above 500. Why 500? I will explain this in a moment. This refers to being just large enough to sustain genetic diversity. The effective population size is a term that not all of you might be familiar with. This is a population genetics terms that refers more or less to how many individuals are contributing genetically to the variation of the next generation. It's not the same that the census size or the number of individuals, because not every individual reproduces, and also because some individuals more similar will produce less genetic difference to the next generation than if individuals that are more dissimilar reproduce. We can estimate the effective population size with genetic data by doing a sampling of a population, extracting DNA, bioinformatic analysis, and estimating NE. This is the gold standard. However, after decades of empirical research and also theoretical, we have realized that the effective population size tends to be around 10% of the number of mature individuals. This varies from taxonomic group, but is from 10 to 30%. That means that if I have 5,000 frogs, adult frogs, then the effective population size should be around 500. And I keep repeating 500. Why? Because, again, from decades of theoretical and empirical research on conservation genetics, we know that below a threshold of 500, populations start to lose genetic diversity. This doesn't mean that they are endangered genetically. It just means that they are losing options to face environmental change. And remember, we are aiming to conserve as much adaptive potential as possible. The fact that we can focus the indicators on processes rather than DNA data itself make it possible to include the genetic diversity assessments as part of other things we already do, like the red listing workshops. For instance, this picture comes from a workshop we have in collaboration with Q and colleagues from Thailand when we are assessing a lot of flower species. And we were able to estimate the indicators from this example that you can see in the screen. So for this species, all of the 4 populations still remain, so the indicator value is 1. But unfortunately, of the 3 populations where we know the size, all of them have relatively few individuals, less than 5,000, meaning an effective population size of less than 500. Doing this kind of assessments, Jess da Silva and I coordinated a large global effort with 9 countries that you can see on the screen, where we assessed more than 900 species, focusing on data that was already available to estimate the indicators for more than 5,000 populations. I'm going to focus on what South Africa learned from this. The pilot study, the 9-country study, was impactful in South Africa because now the genetic diversity indicators feature in their national biodiversity assessment. But they didn't become a national headline indicator because they wanted to have more species. So Jessica da Silva took as a personal challenge to undertake complete taxon assessments as part of the red listing workshops so that they could have more species and there were no issues on why and how some species were chosen. Just very briefly, the kind of results they already have. They assess around 300 mammal species. 96 of them had data on the number of populations. Around of them, 40% had size data. That means they were able to count how many individuals there were to assess any indicator. And they don't really need to have the exact count of individuals. At least to know if there were more than 5,000 individuals was enough to assess the indicator. As for the results, they found that 98% of the populations are maintained. This is good news, though we recognize that there might be some bias in terms of just knowing if a population was extinct or not. For the effective population size indicator, they found that around 68 populations are above the threshold, which means that some populations are becoming too small to retain diversity. These values are similar to what we found at the global level in the 9 countries. Therefore, we think that the indicators are robust. And just to close, the indicators also highlight the importance of local efforts. In this example from Mexico, you can see that the larger population is the one that is being managed and monitored by a local community that is paying attention to this population of an endangered species. So I do believe that conserving genetic diversity will help us to retain the species' adaptive potential, to bend the curve back, and to reverse the red. Thank you very much, everyone. This is the effort of a lot of people, just to close. Thank you.
It's great, nice. Thank you very much, Alicia. Yeah, it makes sense. And if I can take you a bit on the red list fields, we all, well, most of us are well familiar with the red list. It's not only a list of bad news, it's often a system of information with lots of data, as you just showed. I was just wondering, does the genetic indicator could say anything different from the IUCN red listing assessments?
Yes, actually, I have a slide that I didn't show, but this comes from the 9-country paper. So this is gathering data of 9 countries. You can see in dots, black dots, the indicator value for the effective population size indicator. 1 point is a species. So as expected, endangered species have low values, but also we found that less concerned, near-threatened species also have populations that look too small. So when we assess them at the species level, things can look fine, but when you zoom in at the population level, we can realize that populations are becoming too small. This is worrying because they are starting to lose genetic diversity, and we will not see this unless we assess the indicators at the genetic level. Yeah. I hope this
Okay,
answers.
thank you. Yeah. It makes sense. Thank you very much. Interesting figure. Thank you. There is no other questions. I can now call Ricky to join us. And Ricky Gams, you work at the Zoological Society of London. You will be introducing the Edge Index. Thank you very much. You also, go ahead.
Can you see that?
Yeah.
Okay, great. Yeah, thanks, Guillaume. As Guillaume said, I'm Ricky. I work at the Zoological Society of London and also co-chair of the Phylogenetic Diversity Specialist Group with the IUCN Species Survival Commission. Here, I'm just going to give a very kind of large overview into the EDGE concept and the indicator that we've derived from the EDGE concept. And then later on in some talks, we'll dive into the national level examples. So, as I'm sure you're all aware, deep branches across the evolutionary tree of life are currently at risk of extinction, and they're being lost as we speak. Some research a couple of years ago showed that since 1500, we've lost more than 70 tetrapod genera and 10 entire families. And some research we published last year estimated that as many as 900 entire vertebrate genera are at risk of extinction, with more than 50 vertebrate families at risk of extinction. And we also saw that within this data, monotypic families, so those families represented by just a single species, are also disproportionately at risk of extinction compared with your average vertebrate species. So we are currently in this process of pruning these deeper branches of the tree of life. And that's where the concept of EDGE comes in. So what is EDGE? So EDGE stands for evolutionarily distinct, which is the ED part of EDGE, and then globally endangered, which is the GE part. And this was created almost 20 years ago as a metric to prioritize species for conservation action on the ground. And the two main components, the first is evolutionarily distinct species. And these are species that are deemed to be irreplaceable in the way we measure it. And the two ways species are deemed to be evolutionarily distinct is, one way is like the secretary bird here represented on this schematic of a phylogeny. So secretary birds are isolated on the tree of life. They have no close relatives. They're sister to all other raptor species. So this long branch of evolutionary history is solely the responsibility of maintaining secretary birds in the wild. But there's also another way species can be counted or assessed to be evolutionarily distinct, which is where entire lineages are at risk of extinction. So a great example of that is pangolins. So there's 8 species of pangolin in the order. And even though some of them have close relatives, the individual branches that the species represent may not be as deep as the secretary bird. All of the species within this lineage are at risk. So there is actually a real chance that any one of the species of pangolins in the future will be responsible for large amounts of this evolutionary history as we lose species across the tree of life. So we take this evolutionary distinctiveness data and we combine it with our measure of global endangerment, GE, which is simply whether a species is threatened or not and how threatened that species is. So when I say threatened, I mean according to the red list definition in the categories of vulnerable, endangered, and critically endangered. And then for the EDGE index, we're also interested in those species that are extinct and extinct in the wild as capturing their loss as we go. So EDGE scores there combine this idea of irreplaceability of the species with the distinctiveness on the tree of life. And then they combine it with the extinction risk of the species to estimate for every species how much evolutionary history is at risk if we lose that species. And the unit of this measurement is millions of years. So, for example, here we have the galaxy frog. from the Western Ghats. And according to our EDGE estimates, this species potentially embodies 22 million years of evolutionary history at risk if we were not to conserve it. And so we take this set of priority EDGE species forward as, as flagships for conservation and for conservation action and concern. And the idea is that a lot of evidence shows that conserving EDGE species can maintain ecosystem processes. So it can be unique processes to distinct species. A great example of that is the tapir shown here. So evidence has shown that tapirs are quite important seed dispersers in American rainforests due to their unique ability to disperse large seeds that other animals can't consume. And then also evidence shows in birds that ecologically or evolutionarily unique species have ecologically rare roles in networks across ecosystems. And then beyond that, also from the anthropocentric side, EDGE species are also linked to maintaining unique or a variety of benefits that people take from nature, including from plants, and some evidence in birds shows that more evolutionarily distinct species have a wider range of uses by people. And so the EDGE concept started 20 years ago almost, and is now used by a wide variety of organizations, initiatives, in the conservation planning, resource allocation, or even biodiversity monitoring. And to support that, we create EDGE scores or maintain EDGE scores for now more than 400,000 species. So we've assessed more than 70,000 vertebrate species, which we published last year. And a colleague of ours at Kew Gardens is currently in the review process of a paper where we assess more than 330,000 species of angiosperm plant. And the goal currently is we're working towards 2 million eukaryote species that have an EDGE score and an evolutionary distinctiveness score estimated to allow us, as we build better understanding of extinction risk data, we can start to assess more and more understudied taxonomic groups for conservation action. And this kind of global ED data set that we've been building is what underpins our ability to produce the EDGE index at global and national scales. So onto the EDGE index itself. It's currently listed as a component indicator for Goal A in the Global Biodiversity Framework. And simply, the index measures the trends in the conservation status of the most evolutionarily distinct species in a clade. So on the graph here, we have the trends for amphibians, mammals, and birds. And we can produce these trends for amphibians, mammals, and birds because we are able to use the global red list index data to track true extinction risk changes over time. And what we do is we take a set of the most evolutionarily distinct species in a clade, and we monitor the proportion of these species that are either threatened with extinction or have become extinct in the time period that we're monitoring. So here we have the trends for amphibians, mammals, and birds, but we're also working on, you can see for 2023, we have a provisional time point for other vertebrate groups where we're able to estimate their current level of extinction risk across the most evolutionarily distinct species. And I know this is quite counterintuitive if you're used to the red list index, but here 0 is a good sign. That means none of the species here are threatened with extinction that form the group of species we monitor, whereas 100% is bad, so that means a proportion of 1. All threatened, all species in the group are threatened or extinct. So we've started to produce this at the global level, and we have some work that we're putting out now where we've produced national disaggregations of this global EDGE index for the groups which we have the data. So this is to allow kind of a national scale monitoring across all the globally comparable data sets that we have, but also it's also a potential tool for countries where maybe data is lacking on certain groups to extract the nationally disaggregated data to monitor their national set of species. And to support this, we've developed a data portal that you can access at the link below, and I'll put that in the chat. And through this data portal, you can extract EDGE indices and another indicator linked to phylogenetic diversity that we maintain. And you can get this at the national and global level for all of the taxonomic groups across the years that we have it available. But I guess what we're really keen to support with the specialist group is supporting nations to produce the indicator on their own using the data that they have nationally produced. So we've committed to supporting the production of the edge indices through the provision of data that we have, code that we've developed, and just advice and expertise and support where we can. And the way this all catalyzed was through the workshop that we had in Paris. You might hear more about that from Julie, where we were brought together to actually develop the guidelines of how countries can produce a nationally produced edge index. So yeah, a massive thank you to the Natural History Museum in Paris for all of their support with that and really getting this off the ground to have a national level edge index that we can roll out and support countries with delivering. Thank you.
Yeah, great. Thank you, Ricky. The Edge Gang is expanding. That's a very dynamic project. It's very exciting. Maybe I can keep you here another minute. I'd like to ask you, you mentioned the Edge scores, and if I understand that we can build like Edge species list, I suppose, at the bottom of your Edge index. I suppose you could even build like edge species list at the national level, right? And then could this be used to select the species to be monitored with the NE 500, indicator, which I saw from Alicia earlier? Do you see certain limitations to that, or if Alicia feels free to join?
Yeah, I mean, it'd be great to hear from Alicia also, but I think that would be an incredible, streamlining and cohesion of the use of the indicators and the resources. So we would love to see nationally produced extinction risk data, particularly, which that can obviously vary from country to country, to inform national edge lists. So, of course, we have the global red list data and we have the global edge lists, but to see nationally produced edge lists which rely on the extinction risk known within the countries themselves, that could be a really good way of taking the indicator itself and turning it back into a planning tool, which then allows you to direct resources in the country to actually bend the curve of the indicator itself. And then using this, to link it to groups where we need to know more about the NE 500 and see where we need to target population restoration, not just to change the curve of the edge index, but also to improve the score on the NE 500 would be amazing.
Alicia, any thought on that?
Yeah, hello. Yes, I
Okay.
totally agree with everything. And I think it's about leveraging what we already have instead of, working independently. And I think that many of the data that we use, it's similar at some point, or it's the same people, the one who knows it. So if you are doing a workshop for one thing, you could just use the same workshop and do 3 things. So it's a good deal, 3 for 1. Yeah.
Great, thank you very much. Let's move on. The next thing as you've seen on the sequence, we go a bit further on the edge topics. I'll call Seb to join. Yes, and it's always good to have a map to highlight things. Maybe you can share your screen. Here we go.
You can see that?
Yes.
Okay.
Seb, you work at the Royal Botanic Gardens Kew also, and you're going to introduce the Edge Zones work.
Yeah,
Are you ready?
well, thank you. Thank you, Guillaume. And hello, everyone. So before we jump into a few National Edge Index case studies, I thought I would first introduce some of these spatial applications of EDGE and how it's being used to identify priority regions at both a global and national level. Now, just as there are species that are more evolutionarily distinct, there are areas that are more evolutionarily distinct as well. Areas such as New Caledonia for its rare and phylogenetically distinct conifers, or the Seychelles for its endemic zooglossid frogs, or New Zealand for its highly unique parrots. And these are areas that might otherwise be overlooked if we were to consider their species richness alone. Unfortunately, a lot of these areas also tend to be highly threatened, and so there is an urgent need for us to figure out where the greatest losses from the tree of life are predicted to occur, both so we can help guide future area-based conservation and to figure out how effective the existing network is towards this purpose. So in light of that, myself and colleagues have been asking a number of questions about where edge species are, where threatened evolutionary history is distributed, and where these priority locations are found. And this has culminated in a piece of research working first with tetrapod animals, a group for which we have edge data for all the main constituent clades. So we took the data from that study and compiled it with spatial data, giving us information for over 33,000 species, which represents 93% of described tetrapods, 3,000 of which are edge species. And what we find is when we look at their national level richness patterns, edge species richness peaks in Madagascar, followed by Mexico and Indonesia. 75% of edge tetrapods are found exclusively in any single country, and over 50% have a range size smaller than the size of Cyprus. Then, using summed edge scores, we were able to map threatened evolutionary history for all tetrapods, which again reveal patterns that increase towards low latitudes and this time peak in the tropical forests of Cameroon, where there is over 700 million years of phylogenetic diversity that is expected to be lost. Now, these maps are useful, but their value to conservation is limited insofar that we cannot disentangle which of these high-scoring regions are being driven by different assemblages of species. So in light of that, we've been developing over the last few years a procedure to identify priority regions for the tree of life, which we have termed edge zones, and defined here as regions of the world containing large amounts of threatened evolutionary history driven by unique clusters of species. The way this worked is we originally worked with tetrapod data at a grid cell resolution, and the method we elected for involves the spatial complementarity of threatened evolutionary history. In practice, this works by iteratively selecting regions based on their high and unique concentration of threatened evolutionary history, where each new selection is of the region that adds the most new threatened evolutionary history to the overall picture until a chosen threshold of threatened evolutionary history is surpassed. And in this way, it builds up a set of sites with unique species compositions. To ensure the reliability of these regions as priorities, we then repeated this procedure 1,000 times to account for some of the uncertainty in the underlying phylogenetic and extinction risk data. We then grouped contiguous cells, giving us tetrapod edge zones, shown here in red. 25 regions spread across 5 continents, occupying less than 1% of the world's land surface, but together harboring 1/3 of threatened tetrapod evolutionary history and edge species, half of which is found nowhere else. Now, in highlighting these regions, our aim is to showcase those places that are of most immediate concern when it comes to safeguarding the tree of life, whilst also being able to guide conservation organizations like the Edge of Existence program and On the Edge in their regional grant making and their regional campaigns. Unfortunately, almost ubiquitously, these edge zones are under high forms of human pressure as measured using the Human Footprint Index, and they are insufficiently protected, with a few regions like New Guinea and Colombia having virtually no protection whatsoever. Now, a useful application of this approach is that it is scalable to different resolutions and transferable to different taxonomic groups. So through work led by Dr. Ignacio Ramos Gutierrez at the Autonomous University of Madrid, we were able to apply this to flowering plants in the Iberian Peninsula, a region encompassing Spain, Portugal, and Andorra, containing over 5,000 flowering plant species that together comprise over 1.6 billion years of threatened evolutionary history. The study compiled regional spatial data along with global phylogenetic data and supplemented global red list assessments with national red list assessments for endemic species. Then, using that outline procedure, we were able to identify 21 edge zones that together represent 90% of the threatened evolutionary history found in the whole region, with the top 7 alone representing 70% of this total. Again, the protection levels in these sites failed to reach internationally recognized targets, and several of these sites would not be selected if we were to run that prioritization based on a different metric like endemic species or threatened species, due to the presence of few but very phylogenetically distinct species found in some of these regions, such as Edge Zone 8, which contains the top-ranked edge species in the whole region, Gyrocarium oppositifolium, which is a critically endangered micro-endemic. So, to summarize, our hope is that this procedure can be used to highlight areas, phylogenetically important areas, where the failure to protect the biodiversity within would have huge consequences for the tree of life. And our aim is to use this tool to continue to shine a light on new overlooked priority areas at all scales, and as more data becomes available for an expanded number of taxonomic groups. For instance, we might not have the kind of global high-resolution spatial data set available to us for sea plants that we do for other groups, but we do have comprehensive geographic data available through repositories like Kew's World Checklist of Vascular Plants. And we can use this to begin to understand where priority regions are, and perhaps more informatively, where they differ from more well-studied groups such as tetrapods. I think that's probably all there's time for. So thank you very much.
Amazing. I like that last slide. Thank you very much, Seb. I've got a question for you. First, a comment. I think it makes me think and insist that each country should, keeping doing efforts and investing in an efficient information system on species, not only assessments and conservation status, but as good as possible distribution data, as we can see. I guess with the Spanish example, that's where, there is a important point to have like as precise data on the distribution of the most important species. And my question would be, do you see any limit of application at the geographic scales? Is there any thresholds, like a too small country or too small region where you couldn't at the end apply the methodology? Because we just saw the world work you've done, the application at the size of Spain, and we know sometimes it's tricky to apply the same methodology to a very, very tiny scale, like very tiny islands. Do you see any limitation there?
Depends on a few things. Often, if we're talking about potentially groups where there's not a whole lot of existing data, like range maps, like invertebrates or even plants, then often the method that's pushed for these days is being able to use what existing distribution data we have and build species distribution models. And a lot of the time, those predictor variables like environmental, climate, or soil layers tend to be at quite a high resolution. So you can increasingly have a finer scale resolution and still be able to understand where these species ranges are, and then meaningfully turn that into a prioritization. And in fact, you could probably even within a small region, this approach could, just thinking off the top of my head, be used to, if you have an instance where you have population level information, you could use it to actually highlight not just species of interest, but evolutionarily distinct populations of interest as well.
So you could imagine that with a grid of a 1 by 1 kilometers thing.
Yeah. It just depends on
Okay.
the amount of distribution data you have and the
course.
underlying kind of prediction data.
Okay, so again, very important to build up a efficient and dynamic information system on species. Thank you, Seb.
Thank you.
Yes, and next, Julie, you're gonna present the EDGE approach for France, sorry. So please, you can share your screen.
Yeah, it's fine for you for the screen.
Yeah, you could put it like
Better.
Wide. Thank you. Perfect. Carry on.
Okay, nice. Thank you, Guillaume, and hello, everyone. So I am Julie Campana, and I'm a postdoctoral researcher at the Institute of Systematics, Evolution, and Biodiversity, so located in Paris at the National Museum of Natural History. And I'm in charge of the project of the national disaggregation of the EDGE approach in France. And in this talk, I will share with you the issue of the national disaggregation in a country with territories spread across several oceans. In France, we have a general strategy for biology conservation, but each of the French territory has its own reality. As you can see with this map, France is a mosaic of highly diverse territories spread across different continents and oceans. The national strategy is designed for metropolitan France and Corsica, following European regulations. And for each overseas territory, as French Guyana, La Réunion, Adelie or French Polynesia, the national strategy is adapted to local reality of biodiversity and local human pressures. Some of these territories have a rich evolutionary history, with many species holding long phylogenetic branches. And the most striking example is New Caledonia, considered as a global biodiversity hotspot. Applying a national disaggregation of EDGE is at the same time a challenge and a happiness because of the differences of evolutionary histories, of species richness, and pressures specific to each territory. Currently, we are working on a guidelines paper, including recommendations on methods to integrate national data and disaggregate the EDGE index at different spatio-temporal resolutions. Several configurations of data using are possible, but we chose to combine global data on evolutionary distinctiveness based on global phylogenies, as Ricky said, and with national data of extinction risk based on national red lists. Now we are exploring the case study of France, and Alex will present the case study of South Africa just after me. The main challenge for France is to define a national EDGE index reflecting the diversity of its all territories. National red lists are quite advanced for vertebrates and plants, but also for some other taxonomy groups, such as butterflies, spiders, crustaceans, and that in most of the French territories. And this results from an intensive work over these last 2 decades, where there was a high deployment of extinction risk assessments for French species across a wide range of taxonomy groups. So, great news, we can count on national red lists for the national disaggregation of EDGE in France. Our study was based on birds records from 10 territories, comprising highlands and continental areas, as you can see there. We calculated an edge index separately for each of these 10 territories. On this graph, each color point represents the value of the edge index for each territory. The color bars represent the confidence interval, and the different colors indicate the location of a given territory in specific oceans, OC. On the left are the edge indices based on the global red list, and on the right, edge indices based on the national red list. This graph shows four main things. First, the edge indices are higher when based on the national red list than on the global red list, because there are more evolutionarily distinct species threatened or extinct at a national scale than at a global scale. Also, the variation is higher for edge indices produced using national red list. That is probably due by the different number of evolutionarily species categorized as data deficient and not evaluated at a national scale. Also, the edge indices differ across the territories, with, in average, higher edge indices for overseas territories than for metropolitan France. And that is probably due to a lower species richness, a higher isolation in the tree of life, a higher level of threat, which is typical of highlands. And then in the overseas territories, we can observe that age indices are quite similar between nearby territories, such as Guadeloupe and Martinique in the Caribbean Sea, or Seychelles Island and Mayotte in the Indian Ocean. And this may reflect high similarity of species compositions, of evolutionary history, and of threats between these places. Our study shows that the disaggregation of edge at a national level contributes to understand how to consider evolutionary history in biodiversity conservation in several different realities. Indeed, we found that French territories differ in the proportion of highly evolutionarily distinct bird species which are threatened at the national level. For birds, overseas territories are home to the highest proportion of evolutionarily distinct and endangered species, probably due to the evolution in isolation, which is typical of highlands. For understanding if this pattern is common to other taxonomic groups, we will now start the study of flowering plants. Finally, our results encourage us to go further in determining the factors driving variation in edge indices across space and over time. And it is also more challenging because how defining concordant and optimized conservation actions in a country as fragmented and spatially dispersed across the planet as France. All what I have just presented results of a teamwork, of course, and that was started by Dr. Rosalie Pellens from ISAEB and Guillaume Gigot from Pratinat, and including many other people. And the guidelines paper was born from the international workshop that Ricky talked about it, that we held at the museum in Paris last year, and that gathering very different people for very different institutions that you can see in the picture. Thank you very much for your attention.
Thank you, Julie. Thank you very much. Time is flying. So I would just take the chance to thank all experts and local communities in all those overseas territories that are contributing to the knowledge and important data useful for all those studies. And if you agree, we can just we just have a few minutes left to ask Alex to introduce the projects and the edge approach in South Africa. Alex, you work in Sanbi, and please go ahead.
Sure. Can you hear me and can you see the screen?
Perfect.
Great. Sorry, just making sure. Cool. Yeah. Hello, everyone. I'm Alex. I'm a master's student with the University of Johannesburg, working here in South Africa with the South African National Biodiversity Institute on herpetology. And I recently joined this team, or last year, because I was incorporating edge concepts into my thesis. And so my part in this puzzle was to generate a case study on South Africa's edge species, more specifically the vertebrates of our country. So South Africa is one of the world's mega diverse countries, as we are home to 3 global biodiversity hotspots, each of which have vastly different climatic regimes, such as the winter rainfall fynbos, the tropical Indian, subtropical East Indian coastal belt, and the semi-arid shrublands. So it's not surprising that we have extremely high species richness, with a staggering number of bird species, reptiles and amphibians, and mammals. Many of which are endemic to South Africa. Around 50% of our reptiles, amphibians, and freshwater fish are found nowhere else on the planet. As a result of this extreme biodiversity, South African conservationists have pushed to generate comprehensive, nationally produced IUCN risk assessments for the country's vertebrate species. And some groups have even been assessed at multiple time periods, which allows us to not only see how threats are evolving with the landscape, but to assess trends in biodiversity loss and gains. These comprehensive sets of assessments over the last 3 decades put South Africa in a position to report on edge-based metrics with a high degree of confidence and to meet global reporting commitments on nearly all native vertebrates. So the aims of this case study was to generate an edge list and edge index for all of South Africa's vertebrate species. And like with Julie's study, we had nationally produced data, so we could do a comparison between global red lists and nationally produced red lists. To see if there's a substantial difference between the two and see if it's informative to be using national red list. Secondly, because some species have multiple assessment periods, we can also see how the edge index has changed for some groups. In this case, that would be amphibians and reptiles. This 2-part aim allows us to explore both the current state of biodiversity risk and how that risk is changing through time. To breeze through the methods very quickly, we're using a lot of the guidelines that Julie discussed and Ricky mentioned initially, using global phylogenies to ensure comparability with other country studies, and then our IUCN categories. Then we could generate our edge index, and we incorporated data deficient and not evaluated species in the form of uncertainty ranges. And so jumping straight into the outputs of this, here is our vertebrate edge index for vertebrate species. On the y-axis, you can see the edge index. This is measured in proportion. So 1 would indicate that all evolutionarily distinct species are threatened, and 0 would indicate that they're all secured. And so what you can see from the get-go is that edge indices from nationally produced sources tend to show higher edge indexes. And on top of that, they generally have smaller confidence intervals. And this disparity is largely due to the number of species being categorized as data deficient or not evaluated in the global data set, when on a national level, they have in actuality been assessed. So together, these higher indices and the lower confidence intervals suggest that the national data sets are more sensitive to the actual threat status and are more reliable in producing a more accurate edge index for the groups. Now, if we were to change this and look at only the species that are endemic to South Africa, we would see a different picture. If I were to put these placement markers here showing just where the average edge index is now for all species and changes to endemics, you see quite a substantial change. Most groups have a higher edge index. Amphibians move up, so do birds, mammals, freshwater fish, marginally reptiles, and then in contrast, the turtles, tortoises, and terrapins actually have an improved edge index when looking at endemics. But largely, the edge index is higher across nearly all endemic vertebrate groups, meaning that our evolutionarily distinct species are proportionally more threatened when we only look at the endemics. For the groups that we had multiple assessments for, the reptiles and amphibians, here it's a similar graph with the edge index shown here, capping here at 0.5. But on the x-axis, we now have the years. So in 1990, those were when the first reptile assessments took place. And in around 2004, that's when the amphibian ones took place. So right off the bat, you can see that the amphibian endemics, edge amphibians, are the most threatened group at around 0.41. This value indicates that around 41% of our irreplaceable evolutionary distinct endemic amphibians are threatened with extinction. And there's a very similar case here with the endemic reptiles faring worse than all native reptiles. But what you could also see is that the edge index has not changed notably over time for any of these groups, indicating that very few least concern or near threatened evolutionarily distinct species have moved into threatened categories over the last few decades. This suggests that the drivers of extinction were already well established decades ago and have not markedly changed. From a conservation perspective, this is both reassuring and deeply concerning. Reassuring because it suggests that biodiversity collapse has not suddenly accelerated, but concerning because it also shows that conservation interventions, which are actively taking place, have not been effective enough to improve the status of these high priority species. Given that the edge index has remained so stable, this demonstrates the critical need to advance conservation for the most evolutionarily distinct species that are at risk. And so leaning into work more from my thesis, and as you saw with Sebastian's talk, edge can be taken further and visualized spatially. I've termed here, I'm not sure if this has been blocked off by this toolkit at the top, but I've termed this edge richness, which is similar to Sebastian's edge species richness, where you take species distributions of the edge species and map that across the country, highlighting localized hotspots of evolutionary history under threat. And so for reptiles, you see there's quite sparsely spread out distributions, which is mostly indicative of just that there are fewer reptiles. There's about 7 out of the 410 species. For amphibians, however, there you see significant overlap between certain distributions, here in the tropical northeast of the country and in the more temperate southwest. These spatial products can feed directly into protected area planning and help decision makers focus limited resources on the most irreplaceable and threatened areas. Particularly in the Cape Peninsula, where there are as many as 5 edge species found in just these 2 grid cells on the Table Mountain region alone. We can see how fragile and geographically restricted these patterns can be, and in such areas, even small-scale habitat loss could have devastating consequences. And so to summarize what these findings imply for South Africa, the case study shows that when we focus on edge species that are entirely restricted within South African boundaries, the edge index values are higher, indicating that our genetically distinct endemic vertebrates are highly threatened. Stable edge trends indicate that while no new threats are rapidly emerging, all threats remain unresolved. This also emphasizes the need to shift from monitoring to actively addressing underlying pressures. Additionally, what South Africa is quite on top of is producing national biodiversity assessments, which actively inform conservation planners. The previous one that took place in 2018 highlighted genetic indicators as a research gap and something that we should start to report more on. And that's exactly where the edge index is coming in for the 2025 assessment, as it's already been incorporated and will be released with the assessment. And so this hopefully marks the first step in bridging science and international targets with actual ground level localized action. And so what does this imply for the edge index? Nationally produced red list data can fill data gaps left by the global red list, particularly for data deficient or unevaluated species. The edge index is most robust when based on comprehensive, locally validated data. So if available, nationally produced red list data is preferred. The edge index, as of now, is a tool for monitoring the current state of diversity, but when we use it to track edge over time, we can also monitor the success of conservation efforts and whether they are actually reversing extinction risk, which aligns nicely with Reverse the Red's goals. And then finally, other expansions like spatial edge mapping can allow us to integrate this work with protected area planning and prioritization. So in conclusion, this work shows that edge indices are most powerful when grounded in comprehensive, locally validated data. And when this is done, it can be used in global reporting, species selection, and spatial prioritization. For South Africa, this approach paints a clear picture for our genetically unique biodiversity and its trajectory, historical trajectory and future trajectory. Cool. Thanks very much for listening.
Great. Thank you very much, Alex. This shows nicely the potential for work at a national level and all the potential for reporting on this global framework for biodiversity. Thank you very much. Time's flies. I think we've reached the end of our session. Yes, I will have to thank everyone. Thank you to all the speakers and all the attendees. I hope this presentation have clarified these topics and explained the range of possibilities. You have all understood that it is, let me share, I'll just put back the names of everyone here. You can see. Yes, so yeah, I'm sure everyone understood it is a collective effort, and it's all this work depends on collective energies, international cooperation, collaborative work, networks, and workshops. So please feel free to get involved. There must be people in your country or not too far away who are working in these fields, and today's group remains also available, of course. Keep in touch, and some of us will even be at the World Congress to discuss this further. Thank you all, and I think, yeah, we can switch off and move on. I wish you a good evening or a good rest of the day wherever you are. Bye-bye.